Good afternoon, everyone, and good morning to everyone who's watching this from the US, and good afternoon whoever is watching from Europe or Israel. We're very excited to be here. Thank you for joining us today to hear how to develop your disruptive product, a diagnostics product based on our technology. My name is Michal. I'm Strategic Partnerships Director at Newsight Imaging, and I have Erez Lev with us.
Hi, everybody. My name is Erez Lev. I'm heading the Spectral business unit at Newsight.
So what we're going to do is talk a little bit first, introduce the agenda, and then tell you what you're going to hear today. So the first part, we're going to do a short introduction about Newsight and how is a semiconductor company related to diagnostics. We're going to talk a little bit about the schedule right now. Afterwards, I'm going to introduce, and Erez will do a demo of the SpectraLIT, the device you see here. You're going to hear more about it later, and it's magic. Later on, after you see a demonstration, we're going to dive deep into the case study about our medical application, basically the research and everything we have done regarding virus detection. It's important to acknowledge, and of course we cannot disclose everything, but you will be already able to see results and many interesting performance capabilities of our device for medical industry. And of course, the best for last will be how to make your own device, basically the application, how you can take this technology to the practical world and how you can actually develop your own diagnostics device. And eventually, we're going to, of course, open it to Q&A and let you ask questions, and we'll hope that we'll be able to answer all of them. If by any chance we miss a question due to time limitation, do not worry. We're going to leave our communication details, and you'll be able to contact us and ask whatever you like. We actually are looking forward to it. So I think we should begin.
Yeah.
So let's start with the introduction. Newsight Imaging is a semiconductor company. We are manufacturing and developing 3D CMOS image sensor chips for two main applications. One is machine vision, and the other one is spectral imaging. Today, we're going to focus on the spectral imaging, but it's already important to acknowledge that although Newsight is only 4 years old, we're a startup, we already managed to release 4 sensors to the market. And we're collaborating with many global leading players in the machine vision sphere and now also in the spectral imaging sphere. And you can see that we have quite a traction. We're working with Fraunhofer. We manufacture in Israel, Israeli Tower Jazz, which is an Israeli-American fab. We got quite a traction, and we're supported by the Israel Innovation Authority. So, of course, we're not acknowledging everything because it's not relevant to this, but what I do want to say about our traction in the medical world, which is the relevance here today, actually regarding our status and traction in this sphere, in the spectral imaging. We're going to elaborate about it later, but actually the SpectraLIT is already working also with the water world. We signed an agreement with the leading Israeli water company, Makorot, for real-time water monitoring. And after the virus spread, we started to discuss with the medical world, and we successfully seen that we are able to detect viruses, and we're going to show you more of this study later on. But the important and delightful news, as we can already share, is that we did sign an agreement, research agreement with a leading hospital in Israel, and we look forward to continue also doing our research to the coronavirus itself. And in terms of the status, the device is in production. Every time we're improving, and every time you have a later and more advanced version of the development kit you're going to see today, we have already an established roadmap, and we're continue to move on to other medical disciplines. So today we're going to focus on the virus detection, but if you are a diagnostics company interested in other medical sphere, it's very relevant as well. So you feel free to contact us and talk about your use case, and it might be very relevant because we are dealing today with many professors and from hospitals and many diagnostic companies approaching with many interesting medical applications, and we have a very high value proposition to offer to them. So this is the SpectraLIT. I think I'm going to leave the stage to Erez to tell you how exactly it works.
Okay, thank you, Michal. So the SpectraLIT is it's a standalone, it's a lightweight spectrometer. It's based on our spectrometer on chip, which is a very, very little device. You can barely see it. It's about 8 by 8 millimeter chip that is a complete spectrometer featured device. The entire system that you see here is actually the entire system that controls everything which is needed in order to take spectral profile from any substance that is actually inserted inside here. Currently, we are covering between 400 to 700 nanometer, and in the future version, we will be covering 400 to 1100 nanometers. We're going to show you a small clip about how the device is working, so bear with me.
Hi everybody, we're going to be showing you a quick demonstration of how the SpectraLIT is working. We have here the SpectraLIT unit. The SpectraLIT unit has a lid which you have inside a special bay that you can put disposable cuvettes or any other cuvettes with the substance that you want to check. In addition, we have the window-based system that actually controls the entire system. I just put the substance inside, and what I can do a spectral analyze. I'm pressing run. Spectral analyze will actually going to give me the spectral profile of the substance that I put inside the device. I can also do a search, meaning that let's say I have some kind of devices that substances that I uploaded into my database, and I want to make a search inside the database and try to match what I have here to the substances that I have in the database. So I'm going to do a operational mode, which is called search. I'm going to press run. While pressing run, it's going to search the entire database and comes up with the substance that match what I have inside the SpectraLIT. I can of course replace the substance with a different one. Let's say I'm going to put this yellow drink, which I don't really know what it is. I'm going to clear the screen, take a speckle signature just to see what I have inside. And now I'm going to do a search because I want to see exactly what it matched to. And as you can see, it has something that I put inside my database, which is lemon tea. So the system can actually do a complete search of spectral profile with substances that I have already uploaded into the database. I can also do a match. Match means that I'm taking a specific substance that I have in the system and want to see whether what I have inside the SpectraLIT is actually matching one of the items that I have inside. So for example, I'm going to put this lemon tea, I think. The lemon tea shows here. This is the lemon tea. Now let's see if what I have here is actually matching the lemon tea. I'm going to press run and it shows match. Of course, I could put other substances. It's very interesting to see what's happening if I put in a blue substance. I'm going to do a spectral analysis to this substance. I'm going to press run, and what you can see is very low absorbance in the blue area, meaning that this is exactly the color that we are seeing in our human eye. As we explained before, the SpectraLIT is a device that actually gives you absorbance values. So as less absorbance that I have in a specific color, this is actually the color that I'm seeing in my eyes.
So just a little bit more about the SpectraLIT. It's based again in the Newsight NSP 2020. It's a spectrometer on a chip. The SpectraLIT generates vectors with 1024 values, which each one of them represents wavelengths between 400 to 700 nanometer. So this is pretty much the resolution that you're getting. It's able to acquire thousands of signatures and generate absorbance spectra within 1 second. And the data, of course, is being exportable, so it can be analyzed in any external tool that you can find, either by MATLAB, which is what we are using, or Python, or any other tools that you are used to work with. This is again how it looks inside. Really, in a nutshell, you have the material is lighted. This is the process actually. It goes through the NSP 2020. It's being processed in an MCU that is located inside the machine, and it creates the absorbance spectral signatures or spectral profiles. It's good to understand, and it's good to know that the spectral, the quantities of spectral profiles that we can generate in 1 second is about 40,000, up to 40,000, which is a pretty large number if you want to make a very large database and train your database to identify the substance that you're looking for. The device is kind of an IoT device. It's connected to the internet. We will discuss it a little bit later. And in the internet, we have all the machine learning and artificial intelligence features that we are applying into the system. Again, it's portable, it's affordable IoT device. It's great. It's actually a spectral diagnosis device with immediate results. It's based on Newsight proven spectral technology on the CMOS image sensor chips, which we manufacture. The Newsight solution POC and demo are already available for other parallel use cases like water monitoring, alcoholic drinks, and ethanol level detections. And we have successful POC that was conducted in a leading medical center in Israel for tropical virus detection, which is the use case that we're going to be showing you now. The device is powered by AI. It's a bidirectional cloud interface, which allow for firmware updates, spectral profile updates, new profile of upload and uploads to the system from for additional substances or pathogens detections. For example, if you have a new mutation of any kind of virus that needs to be detected in the system that is in the field already, we can upload into your system the new profile of the new pathogens and the new mutation of the pathogens, and your system will be able to detect it as well. There is a constant data training for detection accuracy improvement. We are implementing a very powerful artificial intelligence algorithm. It runs on a database that is keep growing all the time because systems are currently sending data from all around the world, and its result in improved detection and classification. And this is something that is run periodically and uploading into the system the new purified classification. This is a general system architecture. We have the SpectraLIT that is connected to the smartphone, or via Bluetooth, or via USB with the version that we have on the website right now. On the cloud server, we have application and usage management, again, for updates, usage type, if it's for virus, beverage, water, whatever the application is. We have everything which related to registration management, user device registration, license management, the Spectral database and the management of the Spectral database, and of course all the machine learning and artificial intelligence tools which are implemented in our server. It's either our server or the user server. Again, as we said, the system is actually collecting spectral profiles from SpectraLITs that are located all over. The spectral profile is a vector with 1026 values. For the first two values are the device ID and some kind of calibration factor which we are putting inside the machine to make sure that all the spectral profiles and all the wavelengths are being the same on every machine that is spread around the world. This is very important in order to create a unified database. Now we'll go to the case study of the virus detection. The research and the test conducted using the SpectraLIT, the same device that you are seeing here. It's using machine learning and artificial intelligence models that build the profile of infected patients. And the successful results now enable us to distinguish between sick and healthy patients. The tests were made using disposable cuvettes. Each test was done on a serum, basically, 50 microliter of serum that was diluted in PBS to create a 2 milliliter solution. So you can understand that this is a very transparent solution, and yet we were able to create a lot of information out of it. The data sets was samples from dengue negative patients, samples from dengue positive patients. Each of the dengue positive samples had a CT value, which we knew about it, and I will later show what we did with this value. And samples of virus which is called chikungunya, or in a short way, chick. And also with this one, we had we did know the CT value of each sample. So now to the results. What you're seeing here is a very clear classification between dengue negative and dengue positive. And you can see that the cross-validation that we did is almost 100%, which means that there is a very clear distinguishing between healthy and sick serums. This is quite remarkable results. And as you can see, each point represents a sample. So you can understand that we took about 150 samples altogether between that actually combine the negative and the positive. What you can see here is a kind of prediction model. Also here you can see that we have almost 100% predicting capability. So the system, as you can see, is very, very accurate. What we did with the city levels, what you see here in the chart is pretty much the city values that we put inside the system, and you can see that we have a correlative factor R square, which is 0.927, which is an amazing result, meaning that I can put any sample on the chart and I can get in very good proximity the city level. And the city level is also something that helps us afterwards to validate anonymous tests that we did, which we hit 100% in anonymous tests. We also did a test with chikungunya positive and against dengue positives, and you can see that we were able to distinguish between those two samples as well. Again, using the same system, and here we took about 100 samples from all together. Now to make your own device, I will give the stage to Michal.
Yeah, so first of all, thank you, Ellis. I think the medical study, it's very interesting because it shows real capabilities to engage in the medical world. Of course, we're not able to disclose all the research and everything we're doing with a partner, and we hope and look forward to be able to release it to the media soon about this partnership in more details. But I think that we were thinking that just to emphasize the opportunity that medical companies, diagnostic companies, companies in this industry have to see that this technology is very validated for viruses, bacteria, etc. And now we see that the demand is crucial to have something real time that can give you results in seconds. And once you have it using light only, it can give a lot of safety reassurance also to medical staff in any point of care, and even in anyone's homes, in an individual home. That he can get tested and he can know about his health. We can see it affecting other industries like mobility industries. Anywhere you need to be able to know who is sick and who is not. So we see the potential is huge. And that leads us to the business model. So it's important to emphasize that we are not a medical company. We're not a medical device company. We do not intend to sell the final devices, and we do not want to do what you know to do best. We are a sensor company. We're a semiconductor company, and therefore our business model works on B2B. We sell business to business. Of course, we sell licensees agreements, customer support agreements. We are a component maker. So we intend to, this reference design is actually a development kit for you to be able to build your own device. And of course, we, if you partner with us, we're going to support you, and we're going to show you in a few, actually it's the next slide, how exactly are the steps to do it. And we will support the whole process and even after it with the technology, with the software and algorithms, everything that is needed. But again, it's important to acknowledge that we basically sell the components and license agreements for the specific application. So that should be clear. Thanks. And that's exactly the stages. So we try to make it simple, and basically we wrote it simple because it's very simple, the way to engage with us. We work in a similar way with current partners in the other industries and in the medical world. We already started to work in the same way. Basically, the first thing you need to do is to buy this very cool device. You have seen it in the demo, but actually when you will buy it and use it yourself, you'll see it's very straightforward. It's very easy to use. Of course, you get the whole customer kit, manual, all the support with us. I'm gonna show you in a second what it includes when you buy this development kit. But basically, it's very straightforward, and it will be very cool from the first time you would try it, because even before you will try to deal with viruses and medical substances whatsoever, you can already put in the cuvette any kind of material and already see it working by yourself. So that's how cool it is. So that's the first step, and believe me, you will enjoy it, as we all did in the office. So for the first visibility check, that's what you should do. The next step, of course, if you're interested, we can conduct a mutual POC, meaning we can do a POC that is on your defined materials. Basically, you can tell us what's your use case, what's your application, what you would want to find using this technology, and we'll assist to provide all the relevant software and algorithms to have a successful POC. So after you have a successful POC, you can choose to continue to the prototype stage, meaning that you can use our technology and the POC and upgrade it to the next level, meaning we put it in the field. It can be any point of care, it can be an ambulance, it can be a house, it can be in your lab, wherever you will define and want to do so, depending on your application, of course, and we'll support the whole entire process. But the important thing in this stage is basically to be able to see that everything is working in the field that the final device should work. So, of course, when we pass this stage successfully, well, congratulations. It means like everything runs smoothly, and we can decide on a commercial partnership and start selling and the components itself to build the final device and so on whatsoever. So the important things to say here is two things. One, each stage has a no-go, no-go, meaning you have no obligation to continue to the next step if you're not confident or if you're not certain. So our goal is to succeed. We do not want to engage in projects that we believe we have no visibility to succeed there. So we will not engage in something that we are not confident we can do. This is A. And B, we want you to feel confident to work with us and continue to the next step. So feel confident that when you buy the SpectraLIT, it doesn't commit you to anything. You can use it however you see fit, but believe us that we really strongly believe that once you do, you will want to engage further and in the other steps. And of course, the other thing is that every stage has a defined cost, and we can discuss it if you're interested, of course, offline. So that's the surprise. The surprise is that we decided to do an exclusive discount for the webinar participants of the SpectraLIT. You can already find it in our website. You can enter Newsight imaging website in our shop. You can already find it there. And now we have a special discount of $500 US dollars. So that's a discount which is exclusive and valid only for 3 weeks. So be quick to use it. But again, if you don't have PayPal and it's very complicated for you to buy it through the internet, you can always approach us, and we will of course assist you to purchase it in any other way that you feel comfortable to do so. But you have to understand that once you purchase this, you get way more than this very beautiful box. Actually, well, we strongly feel it's very pretty as well. But you get the SpectraLIT, and together, of course, you have included all the software package, full software package included for you to start doing any testing you like. You get even 5 disposable cuvettes to start with, a USB cable, since this version is still using USB. What you have seen regarding the IoTs will be our next version. It will be also available soon. As a startup, we move very fast, so it's going to be available very, very soon. And of course, the first purchase already includes 5 hours of support. So anything you want to do when you open it, again, as I mentioned before, you will get a customer kit and everything that gives you step-by-step what you need to do, how you need to install it, although it's very straightforward. But again, if you feel that something is happening, just to be confident, you can always approach us. You have 5 hours of support included, although we are confident you're not going to need that much to have this thing operated. So thank you for bearing and staying with us so far. We hope it was interesting. It was just, of course, it was just a taste for you to have a little bit of glimpse in what this technology can do in the medical sphere. And we'll be happy to open for a short Q&A. And if you have questions, please write it down. I already see that I have received some questions, and I'm going to address them now. But again, if we don't cover all the questions now, please feel confident to contact us. You see my contact details, Era's contact details. We are very happy to answer your questions about the SpectraLIT, about anything related to Newsight and our technology. We'll be very happy to get questions later, even if we don't cover everything now due to time. So I'm going to start with the first question. Yeah, I see that the first question was, what is the spectral resolution of the device?
Well, the spectral resolution of the device, as we discussed before, you're actually getting a vector of 1024 values between 400 to 700 nanometers. So you can figure out exactly what is the resolution.
Okay, I'm going to go to the next question. How are you planning to check serum of corona patients?
Currently, as we said, we are working with the leading hospitals in in Israel. Actually, is one of the most 10 leading hospitals in the world. It's going to be done in their labs, but again, as we know, the coronavirus, the COVID-19, is not currently located in the serum. But we're going to use other solvent in order to check them.
There is another question, follow-up question to the question before about the resolution. So the question is do we have a figure for the sensitivity of the device? It means that someone saying here that we answered a different question from what he meant. He meant that if we can disclose a figure for sensitivity of the device.
Can you show this question here?
Yeah, you can see it here. Yeah, that's the question.
That's the one?
Yeah.
And sorry for not the resolution, the sample factor, the resolution of the device. Do you have figures of sensitivity of other device? As far as the sensitivity right now, as I said, since we designed the entire chip, we can actually change the configuration of the chip on the fly. We also have a way to create several configurations that allow very, very high sensitivity. So basically, very, very low signal can create some kind of results. But again, in order to get deeper into that, this is something that will require some kind of NDA. But I can assure you that we are getting very, very low signals and create data out of them.
Okay, we have another question here. Again, as Erez mentioned, and for everyone here with the webinar, if you have more technical questions, you want to understand, have a deeper glance in the technology, we welcome you to do so. Some of the questions we can answer without NDA, some of them we need an NDA. So please understand and don't hesitate to contact us.
Yeah, I think we have another question here.
Yeah, we have one before. If we also develop chips for the UV spectrum.
Currently, our next stage is to the infrared and near infrared. I think it's on the plan, but we'll probably discuss it later.
For now, it's not our focus.
Yeah.
So, do we have access to the data that is collected?
Well, this is there's a few ways to access the data. Pretty much, if you're using the device locally, you can access the data locally, of course. If the device is hooked to the cloud, if it's something that is licensed from us, you will get some kind of access to the data or to the results of the data that is collected. Or if it's your cloud, you of course own the data and you can get access to it. But in order for do the experiments and the evaluation, the data is actually collected locally in your computer, and you can access it and export it to any analysis device that you decide.
Okay, so I think that we will finish the Q&A session, and we're going to leave it to the contact information and to the email. Please don't hesitate to contact us and boom us with questions. We're looking forward to it. We're doing it daily, and we think that we have strong value proposition. So please reach us out, whether you're interested to buy the SpectraLIT or to learn more about it. We're here for you, and thank you for joining us, and whoever stayed here today to listen. I see it regarding the recording. We're recording this, and we will release the recording of this webinar later on. And also a final thing that I wanted to say is you're always welcome to follow us on social media. We have many things happening, and we try to keep updated the industries about what is happening in our customers and partners. So you can always follow us on LinkedIn or other social medias on the website, and join our distribution list over the email. And of course, you will be the first one to get updates about the progress of our products. Thank you.
Thank you, Michal.